Good afternoon. It's great to see some new and familiar faces in the crowd. My name is Shanelle, and I am here to introduce you all my dissertation work titled Evaluating the Physiological and Immune Responses of Tanner crab to Hematodinium infection. So Hematodinium is a parasite dinoflagellate that poses a threat to Tanner crab and snow crab in the Eastern Bering Sea. Hematodinium causes a disease referred to as syndrome, alters the appearance of an infected crab, and also eventually results in mortality. So far, two distinct clades of Hematodinium have been identified, clade A and clade B. This study focuses on clade Hematodinium, which is an unclassified genus of Hematodinium distributed across the Northern Hemisphere. So Hematodinium poses several physiologic, economic, and global impacts. Hematodinium infection alters the physical appearance of an infected crab, causing a milky, white-looking heme lymph, as well as the appearance of an infected crab. Infected crabs develop into bitter crab syndrome. This is a disease characterized by a bitter aspirin-like taste in infected crab, rendering that crab unmarketable for consumption. Hematodinium has a very high mortality rate, which typically results in death after 10 to 15 months in post-initial infection. Unmarketable crab causes a loss for the market, which I will talk more about in a later slide. This loss of unmarketable crab drives up the cost per consumer, which is a concern for people who enjoy snow crabs. And increase in Hematodinium prevalence time may be related to environmental changes and could have indication that something much more bigger is happening in the Eastern Sea that we cannot really tell yet. So Hematodinium has been found off the coast and in large marine ecosystems across the globe. Hematodinium this map shows a multi-scale representation of the distribution of Hematodinium across the hemisphere. Hematodinium has been found off the shores of the United States, Canada, France, UK, Russia, and China. So Tanner crabs are native to the Eastern Bering Sea, which is one of the large marine ecosystems of the world. Alaskan fisheries of the Eastern Bering Sea are about half of the marine harvest of the United States. And Tanner crab are an economically important fishery sold collectively with snow crabs that generates millions of dollars annually. Oh, I think you messed up. But now I'm going to move on to the most important question of this presentation. Why do we care? Why is this work important? So this here is a figure from the 2021 Eastern Bering Sea snow crab stock assessment that shows prevalence of bitter crab syndrome or Hematodinium since it was first discovered in the late 1980s. We see several spikes over the last decade. As far as increases in bitter crab syndrome prevalence, the NOAA spike in 2015, and since then, Hematodinium or bitter crab syndrome prevalence has remained above 50% in the Eastern Bering Sea. So, with an expected price of $4.12 per crab and $3.98 per snow crab, estimated annual value of this combined snow crab fishery is estimated to be over $180 million. Hematodinium prevalence remaining above percent. You can now see some of the implications that increase in Hematodinium prevalence have on this economic implication. Since 2019, there has been a substantial decline in the population of snow crab, as a very drastic. This drastic decline in immature snow crab has serious implications on fishery recruitment as well as recovery of this fishery. This decline also suggests a substantial drop in the economic value of this fishery, which could be detrimental to communities that are dependent on this important resource. So now that you guys are familiar about what Hematodinium is and the implications that this disease causes, I'm going to go into the components of my work. So my project seeks to fill in the gaps of knowledge regarding Hematodinium and by answering these following questions. How does Hematodinium evade host destruction? Does Hematodinium cause metabolic changes inside of the infected crab, and how? As well as how does the population differ as the varying levels of Hematodinium infection? So to answer these questions, I came up with objectives. The first research objective is to detect Hematodinium as well as bacteria within the collected crabs. Next is to evaluate the microbial communities of said crabs. Following that, I will be performing differential expression analysis, understand the changes in expression in response to varying levels of Hematodinium infection. And lastly, I'll be performing assays, cytometry to understand the physiological changes of the crab in response to Hematodinium infection. Now I'm going to review the methods that I've used so far. Samples were collected during 2019. They were captured at sea by the 2019 NOAA Eastern Bottom Trawl Survey staff. Collected crabs with Harbor, Alaska, where they were acclimated overnight in a saltwater flow tank. 200 crabs total were sampled. Tissue and blood was collected, as well as recovered from maturity, physical signs of Hematodinium infection, as well as any other physical abnormalities. qPCR, or quantitative PCR, is a highly sensitive DNA-based polymerase chain reaction that detects a specific gene amplification in time. I use a cyber-version-based qPCR assay for detection of the 16S gene, which is specific to bacteria. And I use a qPCR assay specific to 18S, which is good for detecting Hematodinium in our samples. So MySeq is a technique that uses generation sequencing to generate a DNA sequence library of the 16S genes that within a specific sample. These generated sequence libraries are then used to compare to a reference database that is used to identify and categorize bacterial gene species within a sample. So collecting MySeq data will allow me to generate a graph like the one that I have on the slide. This is just an example. And this will allow me to evaluate the microbial community, look at the population structure, as well as figure out the abundance of bacterial genus within the individual. So now I'll move into the results that I've collected thus far. So these two tables are showing the results from my 16S and 18S jar assays. I want to point out that all of my individuals were infected with bacteria, which was unexpected for juvenile crabs. None of the individuals, with mostly having a high level of bacterial infection. Inversely, most of my individuals had no Hematodinium infection, and none of them had a high level of Hematodinium infection. It was to be expected. I finished doing my MySeq analysis using software, but I'm still in the early stages of looking at and visualizing this data. But I was able to generate this refraction curve plot, which actually shows the average species diversity within each individual. And I found something really interesting looking at this plot. So if you notice that there are, for moderate infection of Hematodinium, there was a lower species diversity than individuals that had no Hematodinium or no Hematodinium. That's a little here. And this may be the first indication that there may be a relationship between the Hematodinium infection level and bacterial species diversity. It's very interesting, and I'm excited to learn more about this. In order to complete this project and bring this story full circle, next I'll have to complete my MySeq analysis. I'll perform RNA-seq analysis, which will allow me to perform differential expression analysis and evaluate changes in response to infection at a gene level. And I'll also perform cytometry and metabolic assays to understand the changes in the physical cell counts, immune cell counts, as well as to evaluate any changes in metabolic processes. If time permits, I'll perform EPP to validate those differential expression results. And to elaborate on the findings of the study further, I will compare the microbial community results with crabs found to be infected with Hematodinium in other regions, including rare and blue crabs. I'll also compare the differential expression results from Eastern Brightine tanner crabs to tanner crabs from other large marine ecosystems like Southeast Alaska. And if time did not permit, I'll perform the qPCR validate my differential expression results. Thank you guys for coming and listening to me. Yes.
Nice talk. So you mentioned it was 50% of the occurrence in the population.
Yes, yes.
So how are they detecting that, or how are they quantifying that? What level?
Interesting. Actually, that data is a little bit reliable because they're using visual prevalence data, which is the end stage of infection. Very interesting. And we actually are not sure entirely of the real prevalence of variancy, but we can have an idea of how prevalence is visual data. But now they've been collecting samples. There's also archive DNA samples. It's actually a project A to look at these samples and see if we can detect hematodinium using these archive samples. Yes.
Why do you think the bacterial load was so high for the variancy samples?
Well, this poses some interesting questions. Don't know, but we suspect that there may be a community of bacteria that may be working in some sort of symbiosis with the crab. Some way, we're not entirely sure. We really don't know, because crabs should not have bacteria in their blood. It should be sterile. Have an innate immune system. They don't have an acquired immune system. So it's interesting to see that such young have a bacterial population. That's one of the questions that we seek to understand, and we hope that this result will help us further understand why the bacteria is there. And I think that knowing what kind of bacteria is there will help us to answer that question. Is it a pathogenic bacteria or is it a friendly bacteria? We'll see. Yes.
I was curious about your categories of high, medium, and low bacterial loads. They're qualitative, but
we actually
what are your thresholds for each of those?
Sure. Actually, these are based on this right here is based on hematodinium infection. We also did categorize for bacteria, and we had a specific criteria. I don't have the exact numbers on hand, but we used a paper that had what is a typical amount of bacteria copies of a bacterial gene within a cell of bacteria, and that's what we used to calculate based on our PCR results. How many copies are found in the gene per threshold. And use that to calculate, okay, how many estimate how many bacterial are in the gene.
Because I'm at a table before that was categorizing bacterial load. Yeah.
Yes.
So that's based on a 16S copy number.
Yes. Correct. Yes. Any other questions?